Hey folks, in this video we are going to be talking about protein synthesis. Now, protein synthesis usually happens in two different steps. The first step is something called transcription. And transcription just means writing something down or converting it into your own words. If you've ever taken notes in class, you've transcribed or transcripted something. And with transcription, what we are going to do is go from the process or go from the structure of DNA, and we're going to convert it into something called mRNA, messenger RNA. So DNA is deoxyribonucleic acid. mRNA is ribonucleic acid. So deoxy just means the presence. DNA, there's no oxygen. Ribonucleic acid, there is oxygen. And then the other thing we're going to talk about here as well is translation. And in translation, we are going to go from RNA into tRNA. And the idea is with translation, we are basically just converting between two different languages that allow communication to occur. And that's the basic premise of what we'll be doing in that one there. So to start off this process here, what we need to first talk about is, well, DNA synthesis or DNA replication was the last thing we talked about. And so what we're going to do is something very similar. And we're going to operate by the exact same rules. Let's say that we have a strand of DNA. An RNA polymerase, if you remember, the DNA polymerase was what allowed us to actually copy the strands of DNA. In this case, with transcription and translation, what we're going to have is we're going to have an RNA polymerase come along, and it'll be this large molecule that we'll draw down here. This is going to be RNA polymerase. And what RNA polymerase is going to be functioning in doing is it's going to help us create a copy of a DNA strand. Now, what we need to know is that we are still operating under the same rule as we did with DNA synthesis, which is that we can only go in the 5' to 3' direction. And so what we'll see then is that this template strand, which is what we called it last time, the template strand, we're going to have the coding region from the 3' to the 5'. And what this RNA polymerase is going to do is it's going to start making a copy of this DNA. And that'll happen in the 5' to the 3' direction. Now, what's different is because we're transcribing it into a slightly different language, or transcribing it into something that RNA can understand, what we're going to have is something I call a nucleotide shift. And what that means is we are no longer going to have thymines. Instead, we are going to have U for uracils. So if you had a T in the DNA sequence, that would now no longer be present. So if we had the A-T combination, let's say that nucleotide there is an A, the opposite base pair would be a U to pair up with that adenine there. What will happen is that this will continue until it reaches something called a stop codon or termination site, where at some point in time it's going to tell this sequence to actually stop. Now, these transcriptions that are happening are usually happening on genes, which allow us to make proteins. And so these are usually about 10,000 to 50,000 base pairs or nucleotides in length. Okay, about 10,000 to 50,000 base pairs in length. So now when we generate this structure here, this now gets the term of mRNA or messenger RNA. So we have a sequence that looks like this, and this is known as messenger RNA. What is going to happen then is this messenger RNA is going to be taken, and we're going to move this down a little bit to give us some more space. And once we have the mRNA generated, we are now going to enter the process of translation, where we try to get this into a more usable format. And so what will happen with this mRNA is once we have the 5' to the 3' end, it's going to be binded up by a double ribosomal unit. And so this will be a large ribosomal unit, large ribosome, and this will be the small ribosome. And basically those are going to hold on to our mRNA sequence so that something called translation can start to take place. Now, within this ribosomal subunit, there will be 3 sections. There will be an E section, a P section, and an A section. And the P is where all of the action happens. I'm going to leave it at that and not make any further comments on this. But what will happen is these different sites will occur here, and a tRNA has a weird structure. It kind of looks like this. This is sort of what the structure of it looks like. I can draw that a bit better. Let's clean this up. The structure of it sort of looks like all the way down, out, this, and then up like this, and a circle here. And these are just different coding regions, but the 3 important regions here are down here. There will be 3 base pairs that line up with whatever mRNA sequence is present. So let's say that at this P site right now, let's say that we have a C, a G, and a U, and that was the site. There's a specific tRNA sequence that'll come along here. There'll be a specific tRNA sequence that'll come along and join up in that P site right there, and it'll have the corresponding base pairs G, C, and A. And that will code for a specific type of amino acid, which is the building blocks of proteins. So we go from the tRNA to amino acid, and then those will all get joined up to form something called a polypeptide, which is better known as a protein. And so let's say that we had this exact sequence here. As this reaction's happening, the next tRNA would get loaded up in the A section. Once we have this tRNA with the GCA, we could go back up to this diagram here and look for GCA. And you can see that GCA is present right here, and that codes for an alanine, so an ALA. And so we'll see at the top of this, we'll see a little protein start to be forming, and this will be an ALA. And then as the next tRNA or as the next sequence joins up, we'll start to add another protein onto this, and another one, as DJ Khaled once said. And so there's 3 different steps to this translation process. Part of it is initiation, and it usually starts with a methionine or with an MET. And then the second is elongation. And so what will happen here is that process of tRNAs coming into the P section, the next tRNA loading up into the A, and then once this tRNA has donated its protein or allowed the creation of this amino acid here, it'll slide over to the E section, and then it'll leave, and then it can be reused again. And then the third step is termination. And the termination is one of my favorite guns in Call of Duty Black Ops, which is an AUG, which is also a methionine. So I always remember that AUG is the stop codon, is what it's called. That'll tell us when to stop making proteins and when we need to end the actual creation of this protein. And so this is the process of transcription and translation. We start off with transcription, going from DNA to mRNA, and then we have translation from mRNA to tRNA through 2 different processes here. Hope that made sense. Let me know if you have any questions at all.